how it's found, where you find it. Well, exactly do you do with it? What's the whole like secret behind it? How do you consume it? Well, in a nature survival situation, your best bet would be biting the bark that you extract, or you extracting the bark and making a tea out of it. And more homemade remedies, you can make tinctures and capsules out of them as well. But in a survival situation, you're best off probably making a tea out of it to get the full concentration behind it. So, what is this main compound of interest that provides so much healing properties? It's called salicin. What exactly is salicin? Well, first of all, it's a medicinal wonder. It's still being studied and examined, and more is being found about it each day, and its healing properties. And there's also more interest on the derivatives of salicin as well, as they are believed to provide the same impact as salicin does. Interestingly enough, salicin is a precursor to acetylsalicylic acid, which is the main active ingredient in aspirin. So how does it work? I'll just give a brief explanation, not too detailed. Well, salicin is a B glycoside, and this B glycoside, once consumed, is converted into salicylic acid in the body. The hydroxyl groups that you can see on the salicylic acid react with cyclooxygenase 2, which is an enzyme responsible for inflammation in the body. So once it reacts with it, it denatures it and therefore prevents inflammation and provides pain relief in the process. So where's the whole point of my experiment? What am I trying to find out? Well, my main goal is to find how much salicin is found in different types of branches, and to separate these branches, I categorize them by size and orientation. So branches that are 1 cm in diameter or less were categorized as small branches, and those that are bigger than that were categorized as big branches. But I'll specify like exactly like what I look for later on in the presentation. But in order to meet this overall goal, first I had to accomplish 4 major steps in the process. The first one started off with just separating the bark from the willow tree branch. Then I had to successfully extract salicin from the bark. And once I extracted salicin, I had to purify the willow bark extract. And my final step was to find the percent recovery of this purified form. So a more detailed procedure starts with organizing the branches into the small and large categories. So for this part, I would take like the main branch, that would be like categorized as a big branch, and then the little twigs kind of coming out of it, those are categorized as small branches. Once I had done this, I then had to separate the inner bark from the rest of the branch. So I decided to use not all of the bark, but only the inner bark, because the outer bark contains a lot of tannins and all kind of other compounds and stuff that would add to the mass, but not necessarily contain salicin. So to try and make it all comparative, I chose just to extract the inner bark of the branch. And this added some time to the procedure, as I had to be like careful and scrape off the outer bark and make sure none of the woody portions of the branch were included as well. And also, I had, to make it a nice comparison, I had studied and extracted approximately 1 gram of each type of branch for like each sample. And the procedure that I had seen online, they had worked with about 1,000 grams of bark, but that is way too much to deal with. And just by me, one, I don't think it's really feasible in the amount of time I have and the equipment I have. So I decided to work with a fraction of a thousandth of what they did and make my whole experiment in a micro-sized kind of layout. So once these barks were extracted, the next step was to soak these branches in water, which had to be at room temperature. I could have also used ethanol too, or ethanol water mixture, but they all kind of accomplish the same goal of just dissolving the salicin due to their polar polar orientation of the molecules. And once I had soaked it in water, I then had to try to concentrate these solutions using a rotovap at 60 degrees Celsius. When I went to use the rotovap at 60 degrees Celsius the first time, it hadn't like really wasn't evaporating and condensing properly. And I had even raised the temperature to 70 and 80 degrees, and it still wasn't evaporating as it should. So instead, I had to just go to boiling off the excess solution, but it shouldn't have affected any of the salicin concentration, as salicin should be intact, according to research, intact up to 150 degrees Celsius. So I had to make a slight adjustment in my procedure. Once I had done this part, I then allowed the solution to cool to 4 degrees Celsius and left it overnight, like to cool at this temperature. This caused it like the tannins and other particles in the extract solution to precipitate. And the next day I came and centrifuged this solution at 2100 G for 30 minutes each. And then at the bottom of each of the centrifuge solutions, you have this dark brown tannins that were then removed from the solution. I had to make a slight, everything with my procedures is a slight slight adjustment from the procedure that I adapted them from, because I've been working with smaller smaller volumes of solutions. Because in the procedure day, I said 3000 G for 20 minutes. So, but it all worked out fine in the long run. The last parts of the experiment came with running a chromatography column with the solvent. So once these solvents had been centrifuged and most of the tannins had been removed, the final step to make sure the highest purity was achieved was running a chromatography column. And once this is done, the next step was just to simply dry the solutions and weigh them, and then calculate the percent recovery of salicin compared to the original mass of willow bark. Just to give you a better idea of what exactly I did in that first step and what I categorized branches as, you see the big branch at the beginning, and then you see that like categorized as a large branch is like that main branch, and then the smaller branches are like the little twigs that you can see like branching out from that branch. And with each of these samples, I had separated the bark and the cambium. So the cambium is like that little like section in between the bark and the woody portion. And then once I had done this, I had extracted the bark and cambium to find my willow extract. So results in discussion. Well, from the beginning of the experiment, I kind of noticed once I had soaked the willow bark in water, the little willow bark extracts, which are kind of like smaller in size because I had like cut them up smaller. That's just how they came about. Had a darker color than the big willow branches. But this may not necessarily be due to them having more salicin per se in each of the branches, but rather probably based on how I cut them up. They probably had a higher surface area than the large branches, and therefore diluted quicker in the diluted quicker in the water solution. But you never know for sure. And if I was to repeat this experiment, I would try to probably make sure they had the same surface area. And then, of course, as I mentioned earlier, we had that brown tannins removed in the centrifuge. So that proved to be a successful step, and everything before was successful. And then when we reached the chromatography part, I had troubles with extracting the pure form, and it had to deal with finding the right resin for the chromatography column. And therefore, it's still in the process of finding more the purest form, but what I have right now is still pretty pure. And that solution that I heated and weighed and everything, the results showed that there was a slightly higher mass recovery in the little willow bark compared to the big willow bark, which was 9.6% compared to 8.7% recovery. So not a big difference, not a big enough difference to really kind of say you should prefer one over the other. And since everything was done on a small scale and everything had to be so precise, it's so much room for error that there wasn't a drastic enough difference to kind of say you should go with the little willow bark branches over the big willow bark branches. So, and revisiting like what I did for this experiment and all that I learned from it, first, of course, it's chemistry and we all make mistakes in the lab and sets this back all the time. One of the first things I encountered was when soaking the bark in the water, I kind of like didn't push down all of the bark properly and make sure that all of it was submerged. And I had came back after quite some time and I had seen that the parts of the bark that was exposed to the air had like died, and this thin film of like a dark brown thin film was formed over my solution, and that kind of had to like be discarded right away. So that's one thing to kind of pay attention to if you wish to repeat this experiment. And then also another problem that I encountered, I had a lot of samples that I had saved up and had been ready to centrifuge and purify, but I had left them for a long period of time, not in a chilled environment. And what had happened is bacteria had built up on the surface of all those solutions, so all of them had to be discarded. And it's just a learning lesson that it should be common sense that you should like keep things stored. But after it happened after about a week and a half, like having them sit there. So that was another setback in this experiment. And then of course, as I mentioned earlier, the rotovibe, I have problems with that. And if I could figure out like in the future which temperature to use or which how to adjust the volumes to perfectly use the rotovibe to evaporate them, then it could be more efficient in the future. And then of course, as I mentioned too, the resin selection, that's just for example, for the resins I used, they had not allowed the solution to pass through properly, and it's based on molecular size. And I'm not going to get too in depth into it, but that's a major part of the purification, is just selecting the right resin. And this comes with just trying more and more, because chemistry isn't something perfect. You don't always get the results you want. You have to keep finding new ways to adapt and adjust. So in the long run, why is this all important? Well, first of all, like it goes beyond just survivalist situations. This information can be used in industry, can be used in other areas in which you extract tree solutions and try to find the best part of the tree to take it from without harming the tree too much. But in the future, I would like to probably develop on this project and use a large sample size. But of course, to do a large sample size, you need like a lot of people, a lot of hands, because this of course takes time to separate the bark and run these things and let them sit. It takes a lot of time. And also probably you will have to store branches in the spring. So a major part of this experiment was trying to pick a time when you could get relatively good amounts of salicin in the bark. But in spring, that's when the best time to get it is about 12% higher in the spring compared to the fall time, according to one of the studies that I looked at. So it's about most likely just trying to store branches. The salicin doesn't break down once you freeze it and keep it in the freezer. So if you store samples over time, you could kind of like work through it over a longer period of time. And then another thing I kind of wanted to look at specifically for this tree in the future is probably like how close to a water source like is the tree, like, and how does that affect the amount of salicin found in that tree or branches and different parts of the tree. But of course, all of this comes with having more hands, more time as well.
Hey,
And then
Hyder,
of course,
Oh, sorry to interrupt you, Andre. I was just going to let you know you're up to 449. Did you have a whole lot more you wanted to say?
Oh no, I'm almost at the end. Sorry.
Okay, I didn't mean. I'm sorry. Hey, didn't want to cut you off there, but
Oh yeah.
just trying to keep
Right
there.
I'm about at the end. And
okay.
then, all right.
Yeah, just go ahead and finish what you got.
All right. Yeah. So I wanted to kind of look at how the trees distance to water and everything can probably affect the amount of nutrients flowing in it, and specifically salicin flowing into it. And then this could be expanded on a broader range and just looking at other medicinal trees and how they can be used in a healing method, and like comparing different parts, just like what I did in this experiment. So just to give a summary of what all I went over. Well, first of all, the results, they were valuable. I did see a slight difference, but not a very big difference to say like the bigger branches have more than the little branches or vice versa. So it kind of goes to, it's based on your preference of whether you want to kind of extract the little branches or bigger branches. Me personally, after like doing this experiment for a long time, I found it way easier to extract the bigger branches. The bark kind of like peels off better, and it like just removing it from the woody portion is way easier in the bigger branches rather than the smaller branches. But of course, with the smaller branches, you also have that like smaller surface area. Once you kind of like cut it off, you don't have to cut it as much to like break it down. So it kind of up to personal preference when it comes to that. And just in the future, just trying to make it more pure form of salicin. And once this pure form of salicin is achieved, you can kind of get a better idea, a more precise, more accurate comparison between the amount of salicin in the big branches compared to the small branches. And I just want to also again say thank you all for listening and tuning in. And I hope I was able to help you learn a little more about my project, about basically which branch you should kind of choose in a survivalist situation if you want instant healing. So thank you.
Thank you, Andre. Let's give him a round of applause. Okay. I will go ahead and, since we're at 4:51, I'll go ahead and introduce the next speakers. So next up will be Amy Hyder and Leah Jamerson, and they're going to talk about exhaling, let me pronounce that right, exhaling a breath to foster healing, unnatural dipeptide synthesis targeting Pseudomonas. Oh, I can't say this word. I'll let Leah and Amy pronounce that word. So I will hand it over to them. So let me go ahead and let me pull up your presentation here. Here you go. All right.
Can I make it full screen?
Yeah. Here, let me make it full screen.
Oh, okay.
There you go.
Oh, okay. Yeah. Okay. No, I think they can see me on that.
Can we see that? Is it screen share? You can see the screen from Zoom.
Yeah, because the camera's pointed at the projector.
Yeah. The camera's pointing to the projector. Yeah.
Okay, cool. Well, welcome everyone. I'm really excited that you all are here. And it's really exciting to be able to give a presentation in person again, even if we can only see half of your faces. So we're really excited about that. And it's really nice to not have to worry about being necessarily confined to Zoom, which, speaking of which, I think the camera's up there. Hello to whoever is on Zoom. I know some of Leah's family is on there today. And we hope that you learned something valuable today that you may not have known previously, and that you can share and take away as you leave here today. So what exactly did we research? There's a lot of different pieces to this project, and I want to start out by talking a little bit about cystic fibrosis. Cystic fibrosis is an autosomal recessive condition in which there's a deletion mutation in the CFTR gene. And this causes a accumulation of mucus in the CF lung because chloride is not able to pass through the ion channels in the cell the way that they ought to. And not only does this cause a buildup in the mucus in the CF lung, but it also makes it more difficult for CF patients to breathe, and it also provides a perfect breeding ground for bacteria to accumulate in the lung. There is an acronym known as ESCAPE, and it encompasses 6 of the most dangerous pathogens in terms of antibiotic resistance. And one of these pathogens in particular, which encompasses the P of ESCAPE, is one of the most common forms of bacteria that is found in the CF lung, and it's known as Pseudomonas aeruginosa. Pseudomonas has some interesting and complex evading mechanisms in terms of antibiotics, and that makes it to where that CF patients often will have recurrent and lifelong chronic infections with Pseudomonas. There are 2 antibiotics currently on the market that are commonly used to treat Pseudomonas infections in CF patients, and those are tobramycin and casein. And while those 2 are really good drugs to alternate between once every 6 months or so, in hopes to prevent the bacteria from becoming resistant to the antibiotic, it is of utmost crucial importance that we are making new antibiotics and we're testing them and we're putting them on the forefront of pharmaceutical research in hopes to provide more options for cystic fibrosis patients if they come across a Pseudomonas infection. So what can we do as undergraduate students just doing research? Well, in terms of modern cystic fibrosis research, it really focuses on the use of dipeptides. And dipeptides are essentially small proteins that are made by building blocks of amino acids in your body. And they are used as microbial to fight microbial infections in the body. And these can also be made in the lab as well. And this provides a way to have different variations of dipeptides and then be able to test those and see which ones are the most effective against Pseudomonas. So what you're looking at now are various alkylating and acylating agents that have been used in the past. And as you can see from this graph here, they've all been quite successful so far, particularly the 3 in the middle of the methionine variety, followed by valine and then histidine. And we just marveled at how great that these worked. We were inspired and we thought, why not use other halogen-based alkylating agents to see if they have a similar effect? So that's exactly what we did. We tested the bromine-based compounds and chlorine-based compounds to see how effective that they may be against Pseudomonas infections.
So in order to go about synthesizing these compounds, we first had to start with a resin. So a resin is essentially just a sticky base that we can sort of build like a foundation that we can build the compound onto. So if you look in this figure, it looks kind of scary. It's not. This thing is called a billboard. It has 6 vials in it, and in each vial, we were able to synthesize our unique compound. So we first prepared the resin and put it in each of the vials, and then we added our alkylating agents. So these were the ones that Amy just mentioned, 2 and 3 chlorobenzyl bromide, and then 2 and 3 bromobenzyl bromide. So once we added those, they were in excess, so we did have to, wash the extra away, and that's what it just means by workup there on the screen, because not every bit could stick to the resin. So then we also had to prime this, the alkylating agent, because it wasn't ready just on its own for the new thing to come in. So that was just done by hydrolysis with hydrochloric acid. So once that was ready, then we were able to add our acylating agent, which were our amino acids. So we chose D-valine, D-histidine, and D-methionine. And so we added those in, and of course they were in excess as well. So we washed those away, and it kind of just goes into the reservoir there and then out into the beaker as waste when we do the washing away part. So once they were, our compound was built and our little building blocks, we then had to, cleave it from the resin because we didn't want it to be stuck there forever. So we just kind of pushed it out and into little sample vials. So here, these tables are basically just a 2D representation of how we structured our billboards. So our alkylating agents went in both of the rows, A and B, and then our acylating agents, AKA the amino acids, went into the 3 columns. So that just shows you what sort of each sort of space contained. Here are the structures of all of the 12 compounds we synthesized. They look kind of similar, but they're unique, I promise. So they were unique in which halogen we used, where that halogen attached, and then of course the R group on the amino acids seen in the top left of each structure. Those are unique as well. So of course, then once we had our product made, we had to see if it actually worked. So in order to do this, we had to do something called bioassay. It's a very complicated process. So basically what happened was we took some of our antibiotic that we made and bacteria, and we put it in these little well plates up here and let it incubate for about 22 hours. And so after that, we washed away the extra bacteria that didn't form a biofilm on the bottom of the plate, because that's what actually forms in the CF lung. So we washed the extra away and we actually stained with crystal violet. So that's the purple stuff. It's really pretty in each of the plates. It will stain your whole body though, so just FYI. So then we were able to do spectral analysis with the crystal violet and see like what the concentration of that was. And that was synonymous with the amount of bacteria that remained. So we were able to calculate how much of the bacteria was killed, and then of course how much was not killed. And of course, this was done with our crude product first. We had to filter it, which is purification, and then we did it with our pure product. And then the results that you see on the screen are from our pure products. So this is the control plate that we made. So if you turn your attention to the DMSO here, and then these four on your right. So the DMSO is just the solvent that we dissolved each product in. And then the four on the right are the alkylating agents just by themselves. So we wanted to make sure we tested those with the bacteria to make sure that they didn't kill any, so that we wouldn't have too many variables with that would make us question, was it our antibiotic that killed the Pseudomonas, or was it the solvent or the alkylating agent? And then of course, the other two that remain, aztreonam and the tobramycin. Aztreonam is just a generic form of the casein antibiotic that Amy mentioned earlier. And so ideally, since these are drugs that real life people take every day, we would expect the percentage of bacteria killed to be a little bit higher. But we have a sort of theory as to why that occurred, which you will find out if you just stay tuned. So here are our specific results. The graph with the chlorine-based alkylating agent is on your left, I think. I'm not good with right and left. And then the bromine-based, use my little, here we go, laser pointer, bromine on that side. So as you can see with the chlorine-based products, none of them actually killed any bacteria. They were negative. So -% of bacteria is the same thing as saying bacteria actually grew in the plate. So that was super unfortunate. But we think that the reason that happened is because we were only able to incubate them for about 22 hours. And we actually think that we really needed to do that for maybe twice that, just because we were on a little bit of a time crunch. But so we do think that time is a super sensitive, factor in this specific research as far as how much time the antibiotic and the bacteria need to grow together.
So the figure above Leah is showing from the pure bioassay the bromo-based alkylating agents. And one of the most really important things to take away from this one is the three to your right, the 3 bromo-based alkylating agents. Overall, these were the most successful in treating Pseudomonas infections, and 2-bromo wasn't as much, with the exception of 2-bromo dihistidine. This is really interesting when you look at this graph, because this is showing kind of a contrast. So what you see in orange is the results that we got from our research, whereas those in blue are from the same compounds, but from past experiments. So how we had determined that Chloro was not so effective in treating Pseudomonas infections. Previous research has shown that it is. And once again, this could just be to show that time is really sensitive and the bacteria do need more time to incubate and grow within 22 hours. Once, as Leah said, it could be twice that. And once this is done again, it would be really interesting to see if the results are similar or if they're different based on how long that the bacteria are able to incubate.
So to kind of sort of sum up what we saw as far as trends go, the least successful was not surprisingly all of the chlorine-based compounds. The least successful was 3-chlorobenzyl bromide paired with dimethionine. So that was the one in which bacteria actually grew the most. So that was really just disappointing. But our most successful was one of Amy's that she made, 2-bromobenzyl bromide paired with dimethionine. So that was the one that killed the most bacteria. So we were excited about that. And then just a couple more trends. It was that any bromine-based alkylating agent paired with D-valine and D-histidine, those seem to perform better. And the most specific alkylating agent that worked the best was 3-bromobenzyl bromide. So we were, the future is bright for those things.
So just a quick summary of all of our research thus far. So the alkylating agents that were chlorine-based were the least effective in treating Pseudomonas infections, whereas the alkylating agents that were bromine-based were the most effective. In particular, the 3-bromobenzyl bromide variety was overall the most successful in treating Pseudomonas infections and could be used in further experimentation. There was also a potential for 4-bromobenzyl bromide to see if the results are similar, to see if it's more successful or least successful, and that could be a good thing to test in the future. There's also the chance to re-experiment with the chlorine-based alkylating agents, as we did not have much success with those. It would be interesting to see if there was less of a time crunch, how similar that the results would be to past experimentation. While we don't have all the answers, we may never have all the answers. We are one step closer, and that's one step closer to healing.
Anybody have any questions? We got time for maybe 1 or 2 quick questions. Yeah, Matt.
So I was really interested in hearing about the dipeptides. Did you ever test this, or do you know if they're also lethal or dangerous for mammal cells, like cells that would also be in a human, or lung cells in particular?
So our body produces its own dipeptides, not these specific
Right.
ones, like you're not just gonna find one with a bromine like
Right.
stuck on it. But yeah, so I don't think, that might be toxic, but I'm not sure as far as production, I think, the body certainly does produce them. It's like part of the innate defense system. That's something that we talked about. And so, yeah, I don't have, a specific idea on how, lethal or anything that would be.
Yeah,
Why did you choose the specific amino acids to test that you did?
That's a good question.
that is a good question. But previous research had indicated that these would be successful amino acids to use. So we're just really essentially building on previous research.
Yes, so several of our colleagues here that have graduated have started a database that we're trying to compile information into. So we're just trying to make all kinds of combinations of things and just get a database, like I said, for what could work and potential. We could use other amino acids, of course, other alkylating agents, and then there's promise for all kinds of things. Yes, Danny.
Why do you think that bromine works better than chlorine? That is a good question.
That is a good
Again,
question.
An assumption, once again, could be time. And it could be that time is incredibly essential here. And honestly, that's something that could be researched in the future to see if our results that we got from this experiment are consistent. See if somebody else gets similar results to what we got, or if in fact there was some kind of error on our part.
As far as the organic chemistry part of it, it could be that bromine has a greater atomic mass than chlorine does. So it could just be, you know, a better leaving group or something like that. We're not exactly sure, but of course, you know, like Amy said, future research could definitely, you know, answer that question for us.
All right, let's thank Amy and Leah once again. I'll introduce the next speaker. So Drake Shull, he is going to talk about evaluation of the in vivo and in vitro effects of polyphenols.
Do I
pronounce that
exit?
right? Polyphenols
I
and
think
of course
so.
virgin coconut oil
Oh, good.
on oxidative stress. I'll let you
Do
take it away.
you want the laser
Yeah.
pointer?
I think I got you. Yeah, I got it right in the PowerPoint there. Yeah, that's it. Okay, cool. All good?
Yeah.
All righty.
Hello, my name is Drake Shull, and over the past year and a half here at Milligan, I've been looking at the effects of a specific compound known as polyphenol and how that works in virgin coconut oil to reduce oxidative stress. So 2 years ago, a Milligan alum named Cassie Anderson actually started this project out, and what she did was just look at coconut oil overall and if that reduced oxidative stress. So she did find results that coconut oil does help to reduce oxidative stress. So what I did was build on her research to see what action in coconut oil was helping to reduce that. So before we get started into the results and stuff, we need to know what actually oxidative stress is. So oxidative stress occurs in your body due to the buildup of free radicals. So what free radicals are is that in all biochemical, chemical, and biological reactions, there is an equal sharing of electrons between molecules to form compounds that make up life in general. So as we see here, in the stable atom, the electrons in the outer shell are actually all shared. However, in a free radical, there's an unsharing of those electrons. And what happens is when this unsharing happens, it can cause things in your body to break down. And in these reactions, things known as reactive oxygen species, ROSs, and reactive nitrogen species, RNSs, form. And these can also be formed through like pollutants in the air as well. So what happens is with the buildup of these reactive oxidative species is that a process known as oxidative stress occurs. Now, ROS species are actually beneficial in your body as well. You have in cellular maturation, so as you age, that is caused by ROSs, and also in your immune system, it also ROSs, a small supply of those, help produce important factors to suppress immunity as well. So, but when there's too much of this, too many ROSs and free radicals in your body, damage can occur. And one of the main things is known as lipid peroxidation. So in your cells, you have cell membranes, and these cell membranes are composed of phospholipids. So phospholipids have a polar head and a non-polar tail. And what that means is that the head loves water and the tail hates water. So in this lipid peroxidation, those free radicals actually polarize the tail, taking that non-polar object into a polar object. This causes your cell membranes to start to unravel and lyse, and that is very bad for your body. And as we can see here in this chart, this is all the different things that free radicals can contribute to, such as disease. And it's not just put to one part of the body. Your whole system is affected with free radicals. So lucky for you, though, your body actually has a natural defense system, and these are known as antioxidants. So what antioxidants do is they can act as electron donors or electron acceptors. So they have that paired electron in their outer shell. So what they do is donate it to your damaged free radical. And this creates a stable free radical in the antioxidant form. And these aren't harmful to your body and can be easily flushed out. That then neutralizes your free radical, which in turn lowers your reactive oxidative species in your body. So there's 3 types of antioxidants in your body: preventative, scavenging, and repair. So preventative is kind of self-explanatory. It prevents the ROSs from ever forming. Scavenging breaks down the reactions that form the ROSs, and repair goes in and repairs when the ROSs and free radicals, the damage they've already done. So although your body does make these, they don't make enough of the essential antioxidants needed to combat free radicals and oxidative stress. So it must be taken in through supplementation or through diet. So dark leafy greens such as spinach and kale that everyone loves eating are a great source of antioxidants, as well as dark berries like blueberries and cherries. And this is they're high in antioxidants due to a simple compound known as polyphenols. So in plants, polyphenols act as UV radiation protection as well as to fight against pathogens. So they are derived from a compound known as phenylalanine, which is an essential amino acid that makes up all the proteins in your body. So it is very useful because it can go from a derivative of something your body already has. Now there's 4 different types: phenolic acids, flavonoids, stilbenes, and lignans, and these are just all the different types of polyphenols and derivatives from that. And they do have high antioxidant power as well. So in this study, what I wanted to do was look at the polyphenol content in virgin coconut oil and compare that to 2 known plants that are high in antioxidants, which are blueberries and the anise essential oil. So what I did was first break down those different materials, whether the coconut oil, blueberry, and the oil, and then I did an assay to figure out the total concentration of polyphenols per gram of plant material. And how I did this was I took a gallic acid calibration curve. So gallic acid is a known standard of polyphenol. And by taking that calibration curve, I could take the milligrams of gallic acid per liter of solution that I took, and then convert that to grams of gallic acid and polyphenols in the plant material per gram of plant material. So in your body, we have metals, and metals are useful in our body, such as iron. Hemoglobin helps to carry oxygen throughout your body, and the core of hemoglobin is iron. However, iron exists in 2 different forms, an Fe3+ and an Fe2+. Iron in your body that carries hemoglobin is the Fe2+, but if it oxidizes into the Fe3+, that's harmful to your body. So through a FRAP assay, we see how these plant materials can reduce that Fe3+ into Fe2+. We then looked at hydrogen peroxide and scavenging. So when free radicals form in the body, hydrogen peroxide can form as well, and hydrogen peroxide is not good for your body at all. And then a DPPH assay was performed. And what this basically was is DPPH can induce free radical scavenging. It's just a chemical that induces that. And then we'll find and take that and compare it to how well these plant materials can actually scavenge those free radicals to prevent those ROSs from forming. And the last thing I did was I actually induced oxidative stress into yeast cells. And this was actually building on what Cass did in her project as well, is you first induce you induce your yeast cells with metals, and then in these well plates, you put your polyphenols. I also looked at polyphenols by themselves and the plant matter in general and see if there was polyphenols work better or there's other stuff in plant material as well. And then you induce these and incubate them with your yeast cells either before the metals entered, after the metals entered, or both before and after the metal. And this well plate here is just showing how I set up. I tested 4 metals that are common to the body. So copper, Fe2+, Fe3+, and aluminum. So from the results to the total polyphenol concentration in coconut oil was about 0.0068 grams of polyphenol per gram of virgin coconut oil. And this is a lot lower than the blueberry, which was 0.023 grams per plant material, and the anise oil, which was 0.04 grams of polyphenol per milliliter of oil. And although the amount of polyphenol concentration in virgin coconut oil is so much lower than the blueberries, it still showed antioxidant activity and reducing oxidative stress in the yeast cells eventually that we'll get to. And it's also interesting to note that our FRAP assay, in it, the virgin coconut oil was actually able to reduce more moles of Fe3+ to Fe2+ than the blueberry itself. And I went in and analyzed this, and I think the reason for this is with the blueberry, I had to strain out the juice and just use the juice for the assay, because if I tried to use the whole plant material, the skin and other stuff would get stuck in the pipette and we don't need any more broken stuff in our lab. So I made sure to not break anything else. So that could be a complementation of that. As we can see here, the anise oil had almost a lot higher, almost double what the virgin coconut oil was able to do in reduction power. So the first graph here is our free radical scavenging activity assay, and that was the DPPH assay. So our y-axis is showing how much free radical scavenging was able to happen, and the higher of free radical scavenging activity, the better. And then the concentration of the extract, I looked at 0.05, 0.1, 0.2, 0.4, and 0.8 millimolar concentrations of each plant extract. I did compare this to a known chemical standard, BHT, which is good at scavenging free radicals, and then ascorbic acid, which is vitamin C, which is also known to be very good at scavenging free radicals as well. So at the very beginning, the yellow here is the virgin coconut oil, and it has very low free radical scavenging activity. But as we can see, the trend going up, it is increasing its activity in higher concentrations. So going forward from this and taking these results, if virgin coconut oil is going to be used, it needs to be used probably at a higher concentration than in the body if it wants to help reduce the oxidative stress. So this next one is our percent inhibition of the hydrogen peroxide. So our control was just a known cocktail of scavengers that would be able to reduce that. And then our blueberry and anise oil had high percentage inhibition of the hydrogen peroxide, but the virgin coconut oil had very minimal. So this is very interesting to note because virgin coconut oil is showing great reduction of oxidative stress. However, it's not doing percent inhibition of the hydrogen peroxide. So what this means is that the hydrogen peroxide is usually formed after the free radicals form. So virgin coconut oil is not good at reducing oxidative stress after it's already happened. It has to be before, and it's more of a preventative type of method. So this next graph kind of blacked all of that. We're going to go through each metal by metal because just showing the full graph, as we'll see at the end, it's very complicated and a lot of stuff put together. So our y-axis here is moles of TMB. So what this is, it's hard to actually measure how much oxidative stress is going on in the body or in the yeast cells itself. So by measuring the amount of moles of TMB created, this is done by adding D and TB, which is a known standard. And when that reduces down to the TMB, that is showing that oxidative stress is being prevented. So the higher amount of moles of TMB that form, the less oxidative stress is actually happening in these cells. So our far column here, our dark blue, is the control of just known ROS scavengers. And then we go in order from the virgin coconut oil, just the plant material. And this is before metals was induced to after, before and after. Then we have our polyphenols added. This next huge spike is our blueberries, and then down here is our anise oil. So through these metal trials, we can see that the virgin coconut oil, just the plant matter itself, is able to keep up with the control and does have that reduction in oxidative stress. However, the polyphenols are a little bit lower and they're not really showing much activity at all. However, the blueberry does show in both the polyphenol content and through the regular content that it can reduce oxidative stress by quite a bit. And this is just a trend going throughout. Now, the different metals and Cass research that kind of showed this too, is that they work best on the iron metals, but the copper and aluminum, they're a lot lower. So from this conclusion, I took that the polyphenols do help reduce oxidative stress. However, from those that there wasn't as much activity as just the virgin coconut oil by itself, or just the other plant materials by itself, something else is acting with the polyphenols to help reduce oxidative stress in these cells. So to further this research, what we need to happen is to either break down the polyphenols more and to see their different components, like their structures. Are they a flavonoid or the phenolic acids, and separating those, does something else work, or finding a different compound in the plant as well that can actually help reduce oxidative stress. So before questions, I just want to take a quick moment to acknowledge everyone that's helped me. And that picture of Dr. Whitney was the one on Milligan website, so. But I would first like to thank Milligan University and the 499 research program here for allowing me to do this research. Cassidy Anderson, she started this research, so I'm just kind of continuing hers on. Dr. Michael Whitney for allowing the usage of the bacteria and other stuff in the biolab to get the instruments and to help me run the assays as well. And then my mentor, Dr. Kristen Mudrack, who has helped me a lot on this research presentation and just throughout my Milligan career as well. And I know she's always also kept me to actually get this project done, because there's been some times when I slacked off, so she always kept me on my toes as well. So thank you for that, and thank you for everyone for listening. Any questions?
Thank you. Questions for Greg?
So which type of substance, you said there was preventative, scavenging, and repair, which one do you think would be the most relative for this research? Like, what are we looking for?
Yeah, so I think especially when we saw the hydrogen peroxide, we're looking more at preventative. So stopping the oxidative stress from happening, the formation of radicals before they even go. Now, the blueberries and the anise oil probably has more of the scavenging and the repair abilities, but the virgin coconut oil would be mainly preventative. Yeah.
In the beginning, you mentioned that darker foods are more likely to contain more antioxidants. Can you expand on that a little bit? Because I've heard my wife say this, and I don't believe it. So why is it that darker foods have more antioxidants?
I'm not entirely for sure the exact answer, but if I had to take a educated guess at it, it'd be because the polyphenols and the antioxidants prevent like UV radiation. So it's something to do with the sun. So the darker compounds don't let the sun in as much. So I guess something with the sun and the UV radiation breaks those down, which makes it harder.
Any other questions? Okay, let's thank Drake once again. So we have one more presentation. Unfortunately, she couldn't be here, but she's got a YouTube video. So I'll go ahead and get that brought up. So, yeah.
Somebody's trying to get in.
Oh, somebody's in the waiting room? Where's the waiting room? I don't see it.
Participants down there.
I don't see the waiting room on here. Participants. Let's see. I don't see him. I don't see. Okay, that's cool. That's cool. It was a ghost. Okay. So our next speaker who couldn't be here is Rachel Dove. And I'll go ahead and pull up the YouTube video if I can find it. Ah, here it is. I love the title. Is Burger King playing God? So she's going to talk about GMOs in food products. So I'm going to go ahead and play this video.
Hi, my name is Rachel Dove, and my presentation
Here, let me do my screen share.
The study of genetically modified
There we
organisms
go.
in commercially available soy products. My research mentor was Dr. Whitney. So first of all, what are the benefits of genetically modified organisms? We've heard a lot about GMOs in the news, in the paper, everywhere. It's hard to turn around and not hear about GMOs. So why do we do it? Why do we genetically modify organisms? And first, we can increase crop yield. We can also change the nutrient content of these crops. In soybeans specifically, they have different types of oils in them, and we can select for which oil we want. So we can increase oleic acid or oleic oil while decreasing stearic oil, or vice versa, for whatever we need. So we can change the nutrient content. We can make food healthier this way. We can increase durability. We can make organisms that stand up better with less rain or in more extreme climates. Just can survive a harsher winter, things like that, to help increase yield as well, but it gives them greater durability. They can also self-produce pesticides. So this is done through, this is one example. There's a bacteria called Bacillus thuringiensis, and it releases a toxin that is fatal to this insect and its family. This is the European corn borer. And so researchers found that toxin and they said, all right, what if we could put this into plants? So what they did is they took the Cry5-BT toxin gene out of the Bacillus thuringiensis and they inserted it into the genome of the crop. What this did is now the plant is producing the same toxin that Bacillus thuringiensis produced. So when the European corn borer comes to eat the plant, it dies. It creates its own, it ingests the toxin created by the plant, not by the bacteria, but by the plant, and it dies. So in this way, we're creating our own pesticides. There's a lot of concern about spraying pesticides and insecticides on crops. There's potential ecological concerns, what's the impact on human health, that sort of thing. So we can have crops that are producing their own pesticides naturally so that we're not facing those problems by spraying them on. The BT gene and the toxin it creates is completely harmless to animals, to humans, to anything but the European corn borer and most relatives of it. Other benefits of genetically modified organisms, a longer shelf life. We can make products that last longer. And then we can make crops healthier as well. Fruit can be bigger, shinier, better, taste better, but also can be made healthier. Changing the nutrient content can be used to help create healthier foods. So what are some of the risks? There's a religious concern, and a big one, across our religious groups, not just from a Christian perspective, but the Christian perspective is modifying creation like playing God. If God is a creator, and He's created each and every plant, animal in His design, what does it mean when we try to change it? Do we think that we can do it better than God? What are we trying to do in that sense is a big concern for a lot of religious communities. There's also ecological concerns. Gene transfer is a big one. You can genetically modify an organism, you can plant that crop, but there's always a risk of cross-contamination, cross-pollination to other species. And that has unintended consequences, such as the reduction of biodiversity. That's a huge risk when we take natural selection into our own hands. Not only are we reducing the need for mutations and natural selection and the constant improvement of species, we're also potentially harming levels of the food chain. So if we do the Roundup Ready, the BT modification, then that might take out the European corn borers. Well, if we take out the European corn borers from the ecosystem because we remove their food, because they all died because of the BT toxin gene, then we might be affecting the level up, what eats the European corn borers, the level above that. And at the end of the day, we might have a large change in biodiversity because we've taken out something at the source. So that's a huge concern. There's also the risk of being harmful to small farmers. A lot of times these are big companies that are creating genetically modified organisms. And when they create them, they add a terminator gene in there. What this does is it means that the seeds that are planted are only good for the planting season. They can't be reused. So farmers have to keep going back every year and rebuying these seeds at whatever cost the company sets. They can't save them, they can't reuse them. And that's harmful to small farmers. They're at the mercy of these larger companies that just have profit in mind. That can be a really harmful thing for smaller farmers. So non-GMO project. You've probably seen this label on food sources everywhere. What this is it is a company or project, I guess, that has a long list of what makes something non-GMO verified. And this is, if it's a plant or if it's an animal product, what feed did the animals use? If it's a plant, was there genetic modification here within the same factory, something that like, it is an extensive list of qualifications to get this certification. The non-GMO project believes that we don't know enough about genetically modified organisms to understand their impact on the world and on human health. And consumers deserve to know what they're eating. So they gave this label to verify that something is non-GMO from its source to wherever you're buying it. So my research was kind of based upon some of the tests that the GMO project, non-GMO project would do to ensure that something does or does not have genetic modifications. So my research objective was to test for the presence of genetic modifications in commercially available products through polymerase chain reaction genetic analysis. These are the products I tested. I started with the idea of testing soy products. So I want to test soy products because soy is one of the only genetically modified organisms that is approved for human consumption, not just animal feeds or oils and foods, but soy can be modified and eaten. So on the bottom row here are all soy products, 2 types of edamame, one of which has a non-GMO label, but not the non-GMO project label, it's like an independent label. Tofu, which is soy, and then also Impossible burgers. I got an Impossible Whopper from Burger King to test. It's become much more popular in the past few years to have plant-based proteins to help reduce our environmental impact. It's pretty well known that animals release a lot of methane, cows release methane. So if we can start eating plant-based proteins, we can reduce our animal consumption. We can help reduce greenhouse gases that are produced through that, as well as improving other ecological concerns. So after I tested the soy products, I wanted to test some of these beyond these plant-based proteins. So I got a Beyond Burger, I kept the Impossible Whopper, and then I got this light leg smart sausage, and vegetarian sausage. What's really cool about these is they're all made from plants, but they're made to taste and mimic the feel of meat. And the way that Impossible has done it is they've taken a protein from the roots of soy, and they have modified yeast to create this protein. And this creates a heme flavor, it's like the blood flavor that we like in meat. And so that's how Impossible Burgers, Impossible Whoppers, that's how they mimic the flavor of meat, is through genetic modification of yeast. Beyond does it a little bit differently. They take plants and they don't modify them, but they put them through an intense cooking pressurization process to give it a meat flavor and texture. So the way that I did this was I would grind up each sample in a mortar and pestle, boil it down, centrifuge it, and then I would put it through PCR. All of this was done to extract the DNA from the sample, and then the PCR machine I put in primers that would look for a specific sequence and cut there. And so I could put in my little bit of DNA that I extracted, and the PCR machine would cut it and replicate it so that I could use it for future analysis. My experimental design. 3 trials were completed with the Impossible Whopper, single true organic tofu, private selection edamame, picked sweet farms edamame, and a positive and negative control. All of these, with the exception of positive and negative control, which were an oat and a corn product, were all soy-based. So they all stood risk of being genetically modified in some form or fashion. The positive and negative control, we knew they were both plants, but one of them had been genetically modified and the other was certified that it had not. And after I completed those 3 trials, I completed 1 trial with the Impossible Whopper, Beyond Burger, and the light leg smart sausage, and a negative control. And these are all plant-based protein sources. So here's my results from the first trial. So here is a ruler. So when I load DNA into each one of these wells that I've gotten out of the PCR machine, and the way it works is DNA travels down the gel based on its size. So smaller pieces move faster down. So that's how there's 100 base pair pieces here and 1,000 base pair pieces here. So we bought the ruler, we know these sizes for sure, and then we can measure everything else based on that. So this is the positive control down here. I think my face is hiding it, but it's positive control. This is the plant band. If there's a band found here, we know for a fact that plant proteins were used and that there's plant material in there. So as you can see, negative control has a plant band. Both edamames, the Impossible Whopper, and tofu, they all show plant material, which is good. It's what we assumed. This here is the GMO band. Positive control is positive for genetic modification. So we could then look across and see if there was genetic modification across the board. The only place that showed genetic modification was in the Impossible Whopper. The Impossible Whopper had a very faint but very present GMO band, showing that it was genetically modified. So then in trial 4, once again, we have our ruler. The shell was a little different. It was the same, same material, but it traveled a little bit differently, different size. So that's why the pictures look so different. Here's our negative control. So once again, we can see they're all plant materials, but there's no GMO bans anywhere except for here in the Impossible Whopper category. So this shows the Let My Smart Sausage and the Beyond Burger are both non-GMO. Why is this important? Only one sample in the 4th trial showed genetic modification, only one sample in the 1st trial. It was the Impossible Whopper. This is important, it's consistent with published ingredients of the Impossible brand. The Impossible brand is proud to use GMOs. They believe that through genetic modification, we can better our food sources and better our environment and our world. If genetic modification leads to an easier way to get these plant-based proteins, we can start swapping out meat. We can help save our environment. And they're very proud to use that. 2 samples were publicized as certified non-GMO. It's Beyond Burger and Smart Life Sausage. Both of those showed no evidence of genetic modification. That's important because that's what they said that they were. Unfortunately, just saying that you're non-GMO doesn't always mean that you are. There's always a risk of gene transfer and cross-pollination. So it's important to continue testing and retesting in order to keep that non-GMO project verification label. There's a testing and retesting process similar to this to show that at no point in that product's life has it been genetically modified or come in contact with a genetically modified organism that would affect it. And the Big Street Farms edamame, the simple fruit organic tofu, and the private selection edamame also showed no evidence of genetic modification in their trial as well, which was all what they had published, but none of them had the non-GMO project verified label. So why is this important? What's the big idea here? First and foremost, this research I consider to be a success because I did get results. I got results consistent with what each brand published and what they said, which is good for them, and also good for me to show that it is important to test and retest these materials. If I would continue working on this research, I would perhaps pick a different organism to test, something else that is approved for human consumption, like corn. Or I would look more in depth on what we found in the Impossible Whopper. So we know that we found genetic modification, but what was modified? What was changed there? Another conclusion I came to is the importance of labeling. It is incredibly important that consumers know what they're buying and what they're eating. Non-GMO Project has done a great job of creating a very strict label of, yes, this is labeled, we've certified it, this is great. I think the same should be done for products that are genetically modified. Consumers deserve to know what they are buying. And if they're uncomfortable with genetic modification, for whatever reason, they deserve to know before they purchase. So I guess this all begs the question, is Burger King playing God? They've used Impossible Whoppers and Impossible Burgers that is proud to use genetic modification. So I guess it's up to the consumer at the end of the day, if that genetic modification is meant that the king is trying to play God. Thank you.
Is Dr. Mike Whitney here? I don't see him. Okay. I was gonna say, if you had any questions, we could ask the mentor. I should probably stop this video. All right. Stop the share. Okay. Wow, that's pretty cool. Kind of recursive. Anywho, I guess that's it. Thanks a lot, everybody. Let's give everybody a round of applause again. All right. So, everybody have an awesome day. Enjoy the nice weather. We're almost to the finish line. One more week. Yay. All right. Thanks, everybody.